GCSE. Please tell me if it is working or not. Sorry for the delay. I was just trying to make sure that everything was working. Okay, so let's get started. So firstly, we've got Biology Paper 2 we're covering in this session, and I've got a PowerPoint I've made here for the topic of homeostasis. So let's start with this topic. Feel free to answer in the comments as part of revision. So the first thing you need to know about homeostasis is to define homeostasis. What does it mean? So homeostasis is the regulation of all of the internal conditions inside the body. That's the first mark. And the second mark is in order to ensure that you have a stable environment in response to changes both internally and external conditions. So that's how you define homeostasis. Now, the second thing is automatic control systems. So, automatic control systems are things that help the body regulate the internal environment. These include both nervous and hormonal. For example, there are rings. So I'm just going to try and reload. Okay, hopefully it sounds a bit better now. Just tell me if it does. Okay, so going back to that. Um, okay, automatic. What's going on? So the second thing we need to know is how to define automatic control systems. And what is it? Automatic control system. So an automatic control system is something that helps regulate your internal environment. How long is this? This should be hopefully 1 hour and a half going through the content. Yeah. So automatic control system regulates your internal environment, both the nervous and hormonal communication. So nervous is the chemical messages that go around your central nervous system and your neurons, and the hormonal is from this negative feedback. Is it cutting out again? Let me just try and fix this. Hello? Can you guys hear me? I'm so sorry about this constant cutting off. I think the internet's really bad right now. But do tell me if you can hear me. Okay. Sorry for the delays. Again, if it cuts out, just tell me in the chat. So, yeah, it's going to be 1.5 hour long, and let's just continue. So, the final bullet point for this page is negative feedback. So, what is a negative feedback? So, negative feedback is basically this mechanism in which when something is too high in the body, your body then does counteracts this change by making it low again. And the same thing as when it's too low, it counteracts the change by making it too high so that it ends up balancing out. And the whole aim for it is to keep your internal environment stable. Okay, so that's that page done. Let's move on to the next topic, which is the nervous system. So, the nervous system allows humans to react to our surroundings and therefore coordinate your behavior. So, there's different parts of the nervous system which you need to know. So, firstly, we've got the central nervous system. The central nervous system in vertebrates, which is animals with a backbone, includes your spinal cord and your brain. And in mammals, the CNS is connected via sensory neurons and motor neurons. Now, we have sensory neurons, motor neurons, and also relay neurons as well. But sensory neurons are the neurons that carry the information as electrical impulses from the receptors to the CNS. And then you have the motor neurons. The motor neurons carry the electrical impulses from the CNS to the effectors, and the relay is the middle one. Now, two things you need to know: the receptors and the effectors. So, the receptors detect the change in the environment, detect the stimulus, and the effectors carry out the response. So the effectors can either be a muscle for contraction and relaxation, or for glands to release a hormone and trigger a hormone's release. The final bullet point on this page is what is the role of the central nervous system? So, the central nervous system acts as a coordination center. What that means is that it receives information from the receptors and then coordinates a response, means it decides what to do about it, and then ensures that the response is carried out by the effectors. So let's go through that again. The CNS is a coordination center. It receives information from the receptors, coordinates a response, and makes sure that response is carried out by the effectors. Okay, now we go on to the next page. Synapses and reflexes. So, what are both of them? Synapses are the connection between the two neurons. It's basically the gap in between two neurons in which you have the nerve signal that's transferred across. So the chemicals diffuse between the first nerve through the gap to the next one, and that's going through the synapses. It allows an electrical signal to move from one neuron to another neuron. Okay, now the second point, define reflexes. So, reflexes are rapid, automatic responses to certain stimuli that do not involve the conscious brain in order to prevent humans from being injured. So, let's go through that again. Reflexes are rapid, automatic responses to certain stimuli that don't go through the conscious brain in order to get a rapid response and to reduce the chance of being injured. So, an example could be if someone shines a really bright light in your eye, your pupil will then automatically get smaller, which means constrict. And this means that less light can get into your eye, stopping it from being damaged. Now, firstly, why is it important that humans respond to stimuli? Well, organisms need to respond to stimuli, changes in the environment, in order to survive. A single celled organism can easily respond to its environment, but multicellular organisms need to communicate with each other first to create an overall response. And therefore, multicellular organisms, as they've evolved, they've developed things called neurons um, as a neural communication system. Okay, now we also need know what a reflex arc is. So a reflex arc is just the passage of information in a reflex. So basically, what exactly happens when you're rapidly responding to danger or something, a certain dangerous stimuli? So what happens is, firstly, you detect the stimuli. Firstly, there is a stimuli, the change in the environment. Then you get the receptor cells. The receptor cells detect the stimulus. And this could be, for example, if you've hurt your hand, this could be the pain receptor cells. Then the impulse travels through the along the sensory neuron to the central nervous system, the and it's passed so from the sensory neuron where it's in the CNS spinal cord. Now, in the brain and spinal cord, the CNS, a decision is made as it acts as the coordination center. It receives the information from the receptors, it coordinates the response, and it ensures that this is carried out. So it then decides what is it going to do, the brain and the spinal cord together. And then after it's decided that you should move your hand away, the impulses are sent from the relay to the motor neuron, and the motor neuron, through the motor neuron, it's going to the effectors. So the muscles or the glands. Now, in this case, it could be a muscle, causing your muscle to contract, moving your hand away from whatever's causing you pain. Now, this diagram here, which I got from Save My Exams, is really helpful. I would definitely recommend you using this if you want to break down the reflex arc. So you have stimulus, the sensory neurons, the relay neurons, the motor neurons going through the CNS to the effectors, producing a response. Okay, moving on to the next slide. The brain. Just a quick note here, we're not going through the required practicals in this live stream. We're just going through the content, and then the required practicals will be in a later video. Okay, the brain. So, firstly, let's just look a little bit about the brain. So, what is the brain? The brain is a part of the central nervous system, and it's made up of billions and billions of interconnected neurons. Neurons are all connected together in this brain, and the brain is in charge of all complex behaviors. So, everything complex that you do, that humans carry out, that animals carry out, it's all controlled by the brain. Now, that's quite easy to understand. Running, breathing, sleeping, anything, all coordinated by the brain. Now, there's different parts of the brain that are responsible for different things. So, the cerebral cortex is the main part of the brain. This is responsible for things like language, memory, consciousness, things that require active thought. Now, the cerebellum is responsible for things such as muscle coordination and movement. Now, as we said, the cerebral cortex is responsible for active things you're actively thinking about, like your memory, like your language. Now, the almost opposite to that is the medulla, which is more unconscious activities, such as breathing and heart rate, because you don't have to think about breathing and heart rate, but it's still an activity controlled by a part of your brain called the medulla. Now, they might not necessarily ask you the position of them, but they may show you a diagram in the exam and ask you to point out which part is which. So just remember, the majority of the brain is made from the cerebral cortex. You have the second majority from the cerebellum. You have the hypothalamus. Now, the hypothalamus is shown in this diagram. You don't necessarily need to know too much about it. You just need to know that it controls things like your levels, so your water blood levels and things like that. Now that's the brain annotated. Secondly, how do scientists study the brain? They can see an image on the left-hand side, which has a MRI image of the brain, magnetic resonance imaging. And that's one way that scientists can study the brain. So what they do is you have your MRI scanner, which is this big fancy tube-like machine, and you put the person inside of the machine. It then creates a very detailed picture of the brain structures, and the scientists can then use that to find out what areas of the brain are active when people are doing certain activities. So, MRI is when you get the person to do certain activities and then look at the brain and see what parts of the brain are lighting up, and therefore what parts of the brain are responsible. Now, the almost opposite to that is electrical stimulation. So, in MRI, you're getting a person to do activity and then seeing the effects on the brain. In electrical stimulation, you're stimulating parts of the brain and then looking at that effects on the person by looking at how certain parts of the brain affect your activity. The third way for scientists to study the brain is through studying patients who have brain damage. So if a small part of your brain has been damaged, then the effect that this has on the patient can then be studied, and it can then be viewed as whatever the damaged part of the brain causes to a person. That then means that that part of the brain is responsible for a certain thing. Now, finally, limitations of brain study and what are the problems with it. Well, firstly, the brain is a very complex tissue, so it's very difficult to understand. Also, it's protected by the skull or the cranium, which means it's very difficult to gain access to. Now, also, you don't want to cause physical damage or increased problems to people who are suffering patients or just from normal people. You don't want to cause damage. And finally, um, it's very delicate and the tissue is very soft and you do not want to harm it in any way. Now moving on to the next topic, which is the eye, as well as vision defects. Do tell me if you have any questions or you'd like me to slow down or focus on anything in particular. So firstly, parts of the eye. So right on the outside of the eye, we have the sclera, the sclera. Now this is a tough outer layer and it's just responsible to support the eye and maintain its shape. Then you have the cornea. Now, the cornea is this transparent layer of cells found in front of the eye. And what it does is it has its curved shape, and this curved shape allows light to be refracted and allows us to see. Then you have the iris, which is found on the light blue, almost bulges near the pupil. What the iris does is it controls the diameter of the pupil and it controls it depending on the brightness in order to protect the eye. Okay, then you have the lens. So we talked about the iris, vaguely discussed this. So the pupil is the hole in the middle of the eye, and it just controls how much light enters the eye based on the iris. So the pupil and the iris works in unison. And we'll look a little bit about that in a second. Now, the lens is this thing behind the pupil. Now, a lot of people wrongly think that the pupil and the lens should be the other way around, but no, the lens is actually that purple bit in the middle. And what this does is it focuses the light through a process called accommodation into the retina, which is the receptor cells at the back of your eye, and it ensures that it's sensitive to light intensity as well as color. Now, the shape of the lens, so accommodation, is controlled by the ciliary muscles and the suspensory ligaments, which you can also see on the sides. Now, you also have the optic nerve, which transmits the electrical impulses from the eye to the brain so that we can see images in our brain. Now, let's look at how we can adjust our eyes for brightness. As we said, the pupil and the iris are responsible for this function. So what happens is the reason why we have this reflex, firstly, is that very bright light can actually damage your eyes, and therefore you have a reflex to prevent the damage without having to have a much slower response. So, when light receptors in the retina detect that there's too much light, a optical neurons send chemical messages from the optical nerve to the brain to alarm the brain of this. The brain then sends the messages to the iris. Then what happens is you have these two muscles. You have the circular muscles and the reflex muscles, and these are found in the iris. So when there's too much light, the circular muscles contract and the radial muscles relax, and this causes the pupil to become smaller, reducing the amount of light. Sometimes in the exam, it's called constricting when the pupil gets smaller. Now, the opposite process happens in dim light. This time, the radial muscles contract and the circular muscles wider, so you can get more light in during dark conditions. Now, another reflex is focusing on near objects versus focusing on distant objects. So the eye focuses light on the retina by changing the shape of the lens. So when an object is really close up, this requires more focus. So what happens is your ciliary muscles contract and your suspensory ligaments relax. This then means that your lens is thicker and more curved, meaning that you can focus more. Now, sometimes it's referred to as the lens becoming fatter. Now, to look at distant objects, the circular muscles relax and the ciliary muscles contract, meaning that you are less focused on it because your lens is thinner and refracts smaller. Now let's look at vision defects. So short-sighted versus long-sighted. So what is the difference between them? Long-sighted means that people who have long sight struggle to focus on near objects, whereas short-sighted people, which is a majority of people who have glasses, are unable to focus on distant objects. Now, long-sighted is often called hyperopia, whereas short-sighted is often called myopia. So, with hyperopia or long-sightedness, this occurs when the lens is the wrong shape. What happens is the light does not refract on the eyeball, it actually refracts behind the retina. And remember, light has to refract on the retina for you to be able to see. So if it's refracting behind the retina, this then means that you cannot see the image properly or with enough detail. Therefore, you use a convex lens, which is where the lens is caving outwards. And as the lens caves outwards, this corrects the image. The lens then refracts the light rays so that it focuses on the retina, and this medical term, as you said, is hyperopia. Now, the opposite happens with short-sightedness. So with short-sighted, you're unable to focus on distant objects because what happens is the light focuses in front of the retina, so you therefore have a concave lens which caves in so that you're able to see. And this is again called myopia. Now, in terms of treatment, there's three main things. So the first thing is contact lenses. So contact lenses or glasses, they're both physically wearing the concave or convex or convex lens, and it could be on your eye. The difference is with contact lenses, it could be more useful for activities such as sports. However, it could also mean that you're more likely to get eye infections because you're directly placing the lens on your eye. Whereas with glasses, there is a distance. Then you have laser eye surgery. So with laser eye surgery, this is when a laser is used to vaporize the tissue that is in your retina and your cornea. So if it slims down the cornea, this means that it reduces the power of the cornea, which means that you're more able to see because there's less refraction occurring. Now, a surgeon can precisely control how much tissue the laser takes off in order to ensure that you can see. However, obviously, with any surgical procedure, laser eye surgery, there is a risk of, there is just risks of complications and other things like that, such as infections or the eye reacting in a way that actually makes your vision worse. Now, then you have replacement lens surgery. So, replacement lens surgery is often used for long-sightedness, and it actually might be more effective than altering the shape of the cornea. Because in replacement lens surgery, the natural lens is moved and the artificial lens is then used to replace it, which is made of a clear plastic, and it's then inserted in its place. As it involves work inside the eye, it obviously contains higher risk. Okay, moving on to the next topic. Controlling the body temperature. Now, this is quite a quick topic. So, why does the body temperature, first of all, have to be in balance? Feel free to answer in the chat. So it's really important that the body temperature is in balance because This means that things such as enzymes can work and not be denatured. Now, what we have is something called the thermoregulatory center. Now, the thermoregulatory center is this part of the brain which contains these receptors that are sensitive to the temperature of the blood. So let's look at this diagram here, which is a really good showing of the negative feedback cycle of what happens with your body temperature. So if your body temperature exceeds your normal body temperature, you have your thermoregulatory center, and in your thermoregulatory center, you have your thermoregulatory thermoreg, thermoreceptors, and this is found in the hypothalamus. And the thermoregulatory center is a part of the hypothalamus, and it, as well as, so you have the receptors here, because here the thermoregulatory center acts as a coordination center. And remember, a coordination center, what does it do? It receives information, processes a response, and ensures that the response is carried out. In this way, the thermoregulatory center receives information that the blood temperature is too high by just the blood flowing around the brain, it detects it. And also, you also have temperature receptors in your skin. And these also send messages to the thermoregulatory center in the brain to tell you that your blood temperature is too hot. This then causes multiple responses. So, firstly, increased sweating. Now, sweating carries away hot water away as the energy is carried out as the water is carried out. Now, you also may realize that sometimes sweat tastes a bit salty because ions are also taken out via sweat. Now, you also have your other responses like vasodilation, which just means that your blood vessels dilate, which means they get bigger and move closer to the surface of the skin, causing the heat to evaporate out. Now, you also see your hair, and your hairs may lie flat against your skin instead of forming upwards and become erect, because if your hairs become erect, this traps a layer of heat, keeping you warm. So that's what happens when you're too hot. Now, when you're too cold, pretty much the opposite happens. So vasoconstriction instead of vasodilation of the skin to keep heat. You also have something called shivering, which is a skeletal contraction, which jump starts your body and causes you to get because obviously this requires work and work requires respiration. Therefore, you're transferring energy to the warmer parts to warm the body. Okay, now moving on to the endocrine system. So, hormones. What are hormones used as? So, hormones are basically these chemical molecules that are released directly into the blood. Now, what they do is that they are carried into the blood and only affect certain cells. So, when we're talking about hormones versus nerves, we'll look at this in more detail soon. Now, hormones are produced by various glands called endocrine glands. So, for example, the pancreas produces glands such as insulin. Now, hormones tend to have relatively long lasting effects. Here are some types of glands. So, you can see in the image there's different types of glands. So, you've got the pituitary gland, which is often known as the master gland. It just releases a lot of different types of glands: FSH, ADH, LH, etc. A lot of different glands. Now, they direct the hormones and also cause some hormones to bring about change and bring about other hormones release. Then you have the thyroid, which is found at the base of the neck. So the thyroid gland produces the hormone thyroxine. Now, thyroxine is really important for managing the basal metabolic rate, as well as the heart rate and the temperature. Then you see the pancreas. So the pancreas, it produces insulin, so just one hormone, and that is used for the blood glucose levels. You see the adrenal gland, which is found at the top of the kidney, and this produces adrenaline, the fight or flight response. You've got the ovaries in the female or the testes in the male. So the ovaries for the male The ovaries for the female produce estrogen, which is needed for the menstrual cycle, and the testes for the male produces testosterone, which causes the production of sperm. Finally, the pancreas. Oh, we've talked about the pancreas. Okay, now let's look at hormones versus nerves. Well done, guys. We are just over halfway through, so we've got 1 hour left, and we can try to get through this as fast as we can. So, hormones and nerves have their differences. So, hormones have a slower action. They work on they mainly create a general effect, whereas nerves are very fast and create an effect that's only for a specific muscles. So it's a very precise area, because when you're getting a muscle contraction to, for example, move your hand away from a fire, you're only moving your hand. Whereas for a hormone, your hormones will be released in your whole body, creating a more general effect. So we've got nerves, which is very fast action, very short time it lasts, and acts in a very precise area. Hormones have a slower reaction, lasts for a long time, and acts in a more general way. So if a response is really quick, it's probably nervous, and if it's really long and has a long effect, it's hormonal. Moving us on to the next topic, controlling the blood glucose levels. So, controlling blood glucose levels, how does it work? You need to know the cycle, and this diagram, again, is really good from Save My Exams. So, when you're eating foods with loads of carbohydrates, this then puts loads of glucose, which is a type of sugar, into your body from the in your body as it moves from the blood to the gut. This normal metabolism of cells removes glucose from the blood. Now, vigorous exercise removes a lot of glucose from the blood as well. But there are times when we have excess glucose. And excess glucose can be stored in the liver as glycogen, as well as in the muscles as glycogen as well. The level of glucose in the blood must be kept steady, because the changes are monitored and controlled by the pancreas, and it is one example of a negative feedback cycle. So, when your blood glucose levels increase, say after you've had a meal full of carbohydrates, what happens is the pancreas releases the hormone insulin. Now, as the hormone insulin is released, it then stimulates the liver and muscle cells to take up more glucose, as well as convert the excess glucose into glycogen. Okay, so the excess glucose is stored as glycogen, from soluble glucose to insoluble glycogen. Now, when your blood glucose levels decrease, so in an example of when you're doing vigorous exercise or vigorous activity, this means that you don't have enough glucose for energy. So you have another hormone called glucagon. Glucagon is released again from the pancreas, and this stimulates the liver and muscle cells to convert the previously stored insoluble glycogen back to glucose so that it can be stored. Now you can see on the diagram that it says adrenaline also speeds up the conversion of glycogen to glucose for fight or flight. Okay, now moving on to diabetes. So, diabetes is two types. There's type 1 diabetes and type 2 diabetes. So type 1 diabetes is when your pancreas isn't producing enough insulin. So this can be controlled by having insulin injections, and this is called insulin therapy. So it ensures that therefore the glucose is removed effectively from the blood once your food has been digested, stopping the level from getting too high. It's a very effective treatment, and the amount of insulin that needs to be injected does depend on the person's diet and how active they are. Now, type 2 diabetes is a type of diabetes that comes from risk factors such as obesity. It's when when a person becomes resistant to their own insulin after just abuse to their body. Now, this then means that your body no longer responds effectively to the hormone insulin. Now, this is really dangerous because it could cause the person's blood sugar levels to rise to a dangerous level. And being overweight can also increase your risk of the disease, chronic heart disease, and things like that. So, how do we control type 2 diabetes? Well, you can control it by eating a carbohydrate controlled diet and also by ensuring that you're getting regular exercise. Now, moving on to the next topic, the kidneys as well as kidney failure. So, this is a bit of a big topic. Okay, so key points firstly, urea, ions, and water. So let's look at them. Urea, ions, and water. These are the three key substances that are involved in the kidneys. So whilst we're talking about these three substances, we're also going to be talking about the kidney process. So the kidney process is split into three. You have firstly, ultrafiltration. Secondly, selective reabsorption. And thirdly, osmoregulation. Now, these are kind of bordering A-level terms. However, you do need to be familiar with filtration and selective reabsorption. So, during filtration or ultrafiltration, this means that all of the substances in the blood are split. So, the things that are small enough to fit through the permeable walls of the kidney, which is called the nephron, are things like urea, ions, water, glucose, and gases. However, things that cannot fit, such as red blood cells, white blood cells, proteins such as enzymes and hormones, they do not go through the kidney. So that's the first stage, filtration. Then the second stage is known as selective reabsorption. So selective reabsorption is when certain amounts of each of the substances are reabsorbed into the kidney and reabsorbed back. For example, 100% of the glucose is absorbed back, 50% of the water is absorbed back, 0% of urea is absorbed back because urea is toxic, so we don't want urea. So now let's zoom in to urea, ions, and water. So where does urea come from? So when we have proteins through digestion, proteins are then broken down into amino acids via enzymes. These amino acids are deaminated in the liver, which means that the excess amino acids are converted into fats and carbohydrates. So as this deamination happens in the liver, as excess amino acids are converted into fats and carbohydrates, you have these waste products, and one of the waste products is called ammonia. Now, ammonia is toxic, so if you've ever dyed your hair or done anything like that, that contains ammonia, which is a thing that could damage your hair in the long run. Ammonia is toxic, and your body wants to get rid of ammonia, so it then converts the ammonia into a substance called urea, and after it's converted to urea in the liver, it's then transported to the kidneys where it's filtered out of the blood and excreted from the body as urine. So it goes from proteins, breaking it down to amino acids. Amino acids turn into into the valuable products, fats and carbohydrates, as well as the waste product, ammonia. And then ammonia is toxic, so it's converted into urea in the liver, and then it's converted into urine in the kidney. Now, ions. What are ions and where do they come from? A lot of people get scared when they look at ions, but don't worry, it's not too difficult of a concept. Ions are basically salts. Like you can have sodium ions, just different types of ions that come from when we're digesting food. Now, ions are really important because they help determine the ion water concentration. And if the ion water concentration is wrong, this could cause osmosis to occur, and the osmosis could be in wrong conditions in the body. And because it's in wrong conditions in the body, this means that your cells could start to burst because human cells don't have cell walls, and therefore it's not strong enough to hold the osmosis. So, ions are really important to maintain the water concentration. Also, if you've ever noticed that why your sweat tastes a bit salty, well, that's because it contains sodium ions. So ions are also excreted via sweat as well as urine, and also water. So water comes from normally when we're drinking water, and it's excreted through when we're exhaling, through when we're sweating, and obviously through urination. Okay, so we've talked about the kidney processes of ultrafiltration and selective reabsorption. The final process, osmoregulation. So you don't need to know the term for it necessarily. You just need to know that this stage involves the hormone ADH. And ADH is released from the pituitary gland into the bloodstream, and it's constantly being released in our blood. But what happens is during times where we don't have enough water, we haven't had consumed enough water, this then means that your water content in your blood, because it's low, it makes the kidney walls, the kidney tubules, more permeable, so it holds the water and keeps more water in the body so that your urine is more concentrated, and also there's just less urine. Okay, now moving on to the second half of this topic, kidney failure. So, kidney failure is just practically when your kidney is not working. And what is the dangers of kidney failure? Well, waste substances will start to build up. Now, waste substances such as, water doesn't necessarily that damaging. However, if they build up, however, urea will be very damaging because urea is a toxic substance, and therefore it could control death. And also, the water and ions could also make you lose your ability to control the levels of ions in the water, and therefore cause osmosis and also result in death. So, how do we prevent kidney failure? Well, there's two methods, dialysis and kidney transplant. So, dialysis is just filtering your blood. It has to be done regularly, roughly 3 times a week, and lasting, 3 to 4 hours each time. So, it's very time consuming. What you have is you have this dialysis fluid. Now, what happens is your blood, or the person's blood, is flowing between this partially permeable membrane, and on the other side of the partially permeable membrane is the dialysis fluid, and on your side is the blood. So, what happens is the fluid, the permeable wall, allowing things to go through to the dialysis fluid, is permeable to the waste products. So, the ions leave, the waste substances leave. But the proteins and the useful substances that your blood ought to keep it's too big for, so it stays in your blood. Therefore, that's how dialysis ensures that only the waste products is taken out of your blood. Now, the dialysis fluid has the same concentration of dissolved ions and dissolved glucose as healthy blood, which means that the dissolved ions and dissolved glucose will not leave your blood either. So only waste substances and excess ions will then diffuse across the barrier. So if you're asked in an exam, how does dialysis treatment ensure that useful substances in the blood are not lost? Well, firstly, you can say that for big substances like proteins, like enzymes and hormones, it ensures that it's not lost because it would not fit through the partially permeable membrane pores. And for the smaller substances, like the dissolved ions and the water that the body still needs, it ensures that the dialysis is the same concentration as the blood. And as you've learnt in diffusion, if the two substances are the same concentration, diffusion won't happen because there needs to be a concentration difference, a concentration gradient. Okay, so that's dialysis. Um, some people may get fed up with dialysis because the waits between the dialysis means that you're just building up all of these toxic substances and you can't really release it, and it also leaves you really thirsty. So some people might just opt for kidney transplants. So kidney transplants are actually a cure, but the danger is that they can be rejected, and also it needs to be a matter of convenience because somebody would have had to be an organ donor and also have died recently or just been willing to give their kidney so that it could be used. Um, Now, however, there is a small risk because as a person who donates it, the donor kidney could be rejected because as with anything, the body can reject anything if there's something else living inside our body. And if it's someone else's living kidney, our body may recognize this as foreign, just how we recognize, a virus as foreign and expel it out of our body. However, in the long run, transplants are also cheaper. So there's benefits and disadvantages. Now moving on to the menstrual cycle, hormones, and controlling fertility. Well done if you made it this far or if you've just joined. Hi. And if you have any questions, feel free to put them in the chat, or if you would like me to slow down. I'm also going to be doing chemistry live stream and physics live stream today, so feel free to join those if you are available. Okay. Menstrual cycle, hormones, and controlling fertility, part 1. So, defining puberty. What is puberty? So, puberty is when your body, either female or male, starts releasing these sex hormones. And what these hormones do is they start triggering these secondary sexual characteristics, such as, pubic hair, facial hair, your growing of breasts, and things like that. So, in men, the main reproductive hormone is testosterone, and it's produced by the testes in order to stimulate sperm production. In women, the main reproductive hormone is estrogen. It's produced in the ovaries as well as using as well as creating eggs for the menstrual cycle. It's also required to create the physical changes in the girls, as you can see in the right hand image. Now, 4 stages of the menstrual cycle. Key things you need to know. So, in stage 1, which happens on day 1, this means your menstruation starts. So, as you can see, in stage 1, this means your uterus lining starts to shed and break down. This lasts normally 4 days. That's why a period lasts roughly 4 to 7 days. Then in stage 2, the uterus lining is building back up again. Now, as the lining is building back up again from days 4 to 14, this means that you have this thick, spongy layer of full of blood vessels, and they're ready to receive the fertilized egg. In stage 3, an egg is developed and then released, and this happens at day 14. This is called ovulation. And then at stage 4, the wall is then maintained for about 14 to 28 days before it's then broken down again. So just going over that one more time. Stage 1 is day 1, menstruation starts, the uterus lining breaks off. Stage 2, the uterus lining then builds up again as a thick, spongy layer full of blood vessels builds up to receive a fertilized egg. Stage 3, an egg develops and is then released. And stage 4, the wall is then maintained for another 14 days before it's broken down again. Okay, now the final bullet point for this page, 4 hormones in the menstrual cycle. What are they? So the first one is FSH, follicle stimulating hormone. Now, as it may suggest by the name, what happens is this stimulates egg maturation, so allowing eggs to mature. And the second thing it does is it also causes and stimulates the ovaries to produce the next hormone in the cycle, estrogen. So you have FSH, it causes egg maturation and stimulates estrogen. Now, estrogen is produced in the ovaries, and this causes the lining of the uterus to grow. And as the lining of the uterus grows, it stimulates the release of LH, luteinizing hormone, as well as stopping the release of FSH, so eggs stop maturing. Then you have LH, luteinizing hormone. This causes ovulation. And then you have your final hormone, progesterone, which is responsible for the second half of the cycle in which the uterus lining is built up, ready for if an egg was to be implanted. Now moving on to the second half of this topic, which is all about contraception, hormones, and hormones to increase fertility. So, contraception and hormones can involve things without hormones and things both with hormones. So, how can hormones be used to reduce fertility? Well, as we've saw in our last page, both estrogen and progesterone reduce FSH and LH, which means it prevents eggs from even maturing and prevents eggs from ovulating. This then means that if an egg is stopped from being released, then it can't even be fertilized in the first place. So, therefore, if you have ever seen the contraceptive, the pill, lots of people take this daily, which just increases the estrogen and progesterone, which means that your body has so much that egg production just stops. Now, another contraceptive method which contains hormones is things like the contraceptive patch, which contains estrogen and progesterone. This is like a patch that's just put on the arm, and normally it lasts a week. You also have the contraceptive implant. This is inserted under the skin of the arm, releasing a continuous amount of progesterone. This can last for 3 years. Then you have the injection, which contains progesterone for 2 to 3 months. The intrauterine device. So you can see there's so many hormonal methods. In terms of non-hormonal methods, there's things such as the barrier methods, so condom, diaphragm, also the female condom as well. And there's also a thing called spermicide, which is actually a chemical method. So spermicide just kills the sperm, but it's not 100% effective because it only kills roughly 70 to 80% of it. Now, there are more drastic ways as well, such as, vasectomy and female sterilization. So, both of these are sterilization, which involve cutting or tying the fallopian tubes to prevent pregnancy. However, there is even a very small chance that it could just rejoin and you could get pregnant again. Then there's natural methods such as abstinence, which is probably the best contraceptive method because if you're not having sexual intercourse, you're definitely not going to get STIs or anything like that. And there may be the rhythm method as well. Monitoring body temperature is not as common of a one, but yes. Okay. In terms of controlling fertility and increasing fertility, hormones are also used in things like IVF. So they can be used to increase the fertility. FSH and LH tend to be used. So the pros is that it helps a lot of women get pregnant. However, cons is that it doesn't always work. IVF can be used to help couples have children. So what you do is you take out some eggs from the mother, from the woman's ovaries. You fertilize them using the father's sperm. Now, sometimes if the man has a low sperm count, you just inject the egg with the sperm to ensure that it definitely has to start dividing. The fertilized eggs are then grown as embryos in a lab incubator. And once the embryos are tiny balls of cells, known as zygotes, they are then transferred into the woman's uterus, so one or two of them to ensure that you increase the chances. Now, advantages is that they can give an infertile couple a chance of a child. However, disadvantages is that it could lead to multiple births, which could risk the life of the mother or the child or children. And the success rate is actually really low of IVF. In the UK, the success rate of IVF is only 26%, which is really low. And this could then be very stressful for the couple and parents. However, there are advancements being made in microscope techniques which have helped to improve the success rates, micro tools, things like that. Some people obviously are against IVF because they believe, what about the unused embryos? Do they just get to die? Is that unfair? Sanctity of life argument, all of those things. Therefore, some people have some ethical issues with it. Now moving on to the next topic, which is adrenaline and thyroxine. So, adrenaline is a hormone released by the adrenal glands, and it's the fight or flight response is part of that. So during a stressful or scary situation, what happens is your brain detects this fear or this stress and sends these nervous impulses to the adrenal glands. This then responds by secreting adrenaline. It gets the body ready for this fight or flight response by triggering mechanisms that increase the oxygen supply or the glucose supply. Okay. Now, hormone release can also be affected by negative feedback, as we discussed earlier. So then thyroxine. So thyroxine is used to regulate metabolism. It's released in the thyroid gland and plays a really important role in regulating the basal metabolic rate. So if you ever ask what is the basal metabolic rate, this is the speed at which the chemicals reactions in your body are happening when your body is at rest. Now, thyroxine is very important for loads of processes in the body, such as stimulating protein synthesis, growth and development, etc. Okay, now moving on. Plant hormones and commercial usage. This is the final topic in this first topic. Okay, so auxin. So auxin is a plant hormone, and auxin controls the growth of tips near the shoots and roots. So what it does is it allows for positive phototropism as well as gravitropism. So in phototropism, auxin culminates and groups on the shadier side, causing growth in the shadier side so that the plant can get more sunlight. A similar thing in gravitropism, it goes on the lower side of the root in order to push it forward to prevent the roots from growing sideways and to allow the roots to grow towards the gravity. So, what it does is physically, as the auxin moves backwards, it promotes cell elongation. So, that's the key thing you need to know. How does auxin work? It promotes cell elongation. Okay. Now, right about now, there would be a required practical, but we're not covering that in this video. So let's just move on to the 3 uses of auxin. So firstly, auxin can be used for killing weeds, because what happens is you may be like, how can auxin be used for killing weeds if it's used to promote growth in plants and shoots and roots? Well, what exactly happens is that a lot of auxin is put into the plant, and then it then just causes the plant to overload. And because there's so much auxin, it then just dies because it's too much. The rate is too much in comparison to how it should be growing. Now, 2 other uses for auxin include rooting powder for plant cuttings, so just dipping plants in rooting powder, allowing it to grow, and also growing cells in tissue culture. So if you wanted to make copies of really rare plants, then you could just do that as well. Now, gibberellin. Gibberellin is another type of plant growth hormone. It stimulates 3 key functions. So, firstly, it controls seed dormancy. So, lots of seeds won't germinate unless they've been through certain conditions. So, if you add gibberellin, it means that the seeds can reduce their dormancy and quickly grow. It can also induce flowering and induce growing larger fruits. So, inducing flowering, because normally plants require certain conditions to flower. However, gibberellin um, allows the plant to grow, not only start growing flowers in a condition where they might not normally or naturally, but also allows them to grow bigger flowers. And the same thing is with fruit. So it allows plants to grow larger fruit. Now, ethene is another hormone, and what it tends to be used for is commercially used, and it's used to promote the growth of or speed up the ripening of fruits. So, what happens is ethene is this gas produced by aging parts of a plant, and it promotes cell division. So, as fruits, imagine mangoes being delivered from tropical countries and climates to the UK, it stops the ripening by taking out the ethene, and then it allows the ripening despite it being cut off from its original plant in the UK by adding ethene. Now that is the end of the first topic. So well done for making it all the way through. We're going to have a quick 5-minute break as I prepare the next topic, inheritance, variation, and evolution. Unfortunately, there's not a nice PowerPoint like I've made like this, but I'll try to find a good resource. Okay, so we will have a 2-minute break, coming back to you very soon. Hello everyone, just returning back from our break. Hope you had a nice break. We're now going on to our second topic, which is inheritance, variation, and evolution. This topic is much more condensed than the first topic, so don't worry, it shouldn't take a full hour. Hopefully this should take at least probably 15 to 20 minutes, and then we'll go on the last topic as well. If you have any questions, do comment them, but let's get started. So just if you wanted to know this resource, this is on Physics and Maths Tutor. They have these notes for the whole topic, which you might find helpful if you don't like your notes. So let's begin. So the first thing that it talks about here is sexual and asexual reproduction. So let's just see where it refers to DNA and genes, because it makes more sense to start from DNA and genes, I think. Okay, let's start from the DNA structure. That makes more sense. So, DNA stands for deoxyribonucleic acid. I don't believe that they will ask you what DNA stands for, but it's always good to know what it stands for. So, DNA is the chemical that contains the genetic material in a cell. So, when you're talking about genetic material in a cell, you're talking about DNA. That's the chemical we're referring to. Now, it contains this coded information that basically has all of the instructions to put an organism together. So, it's what's in your DNA that determines what inherited characteristics you have. DNA is found in the nucleus of animal and plant cells. It's found in really long structures which are referred to as chromosomes. So this might be easier to just do on a diagram. Let me just quickly get the diagram up. So we've got DNA, and DNA are these chemicals that genetic material is made from. And then you've got chromosomes. So chromosomes is what DNA is split up into. Okay. Let me. This might make it a bit more clear. Okay. So we first have DNA. Deoxyribonucleic acid. So as you said, DNA is the chemical that all genetic material in the nucleus is made up from. So the chemical that all genetic material is made up from. Now, the DNA contains all of these coded information that has all of the instructions needed to put an organism together. So whatever is in your DNA controls whatever inherited characteristics that you have. So DNA is found on these really long structures called chromosomes. So chromosomes normally come in pairs, and DNA is a polymer made up of two strands, and these two strands coiled together form a double helix. So you have chromosomes, which is made up of DNA, so long strands of DNA. So chromosomes are these long strands made of DNA. DNA are the chemicals that all genetic material is made from. Now, DNA, as we said, is what we call a polymer. So a polymer is a long repeated unit chemical. So why we call DNA a polymer is because the DNA is made up of two strands, and these two strands are each called a nucleotide, and they then form this structure which is referred to as the double helix structure. Okay. Now, a gene is a small section of DNA. So DNA is made from chromosomes and genes is made from DNA. So let's look at it that way. So a gene is a small section of DNA that codes for a protein. Or that codes for a particular characteristic. So some people even go further and say a gene is a small section of DNA found on a chromosome, but you don't necessarily have to say the found on the chromosome bit. So we've got, just to review, we've got chromosomes. And chromosomes are long strands of DNA. Then you've got DNA. DNA is the chemical that all genetic material is made from. And then you've got genes. So genes are sections of DNA that code for a protein. Now, genes simply tell us what order we put these amino acids together because there's 20 amino acids. So we don't need to define amino acids. Amino acids are just proteins. Now there's 20 different types of amino acids. Now what genes do is genes determine the order of these 20 different amino acids. And the order is really important because depending on the order is all the different characteristics we get. Now, the DNA also determines what proteins the cell produces. Now, why this is important is because it could produce things like hemoglobin, which could make it a red blood cell. It could produce things like keratin, which makes it another type of cell. Now, this then determines what type of cell. So the DNA is also integral in determining what type of cell we have. Now, every organism has what we call a genome, or a genome, some people call it. Now, either way, no matter how you call it, we've got the word almost gene in it. And what that means is just that the genome is the entire set of genetic material in an organism. So, the entire set of genetic material in an organism. So what does that mean? What does the entire set of genetic material in an organism mean? So when we're talking about a human genome, that refers to everything from the genes for your eyes, the genes for your hair, the genes for both your physical and your mental characteristics. Now, why is understanding the genome important? Why would it be really beneficial? Well, if we understood the human genome, and as scientists try to, as we do understand it, it can also help us identify genes that are linked to different types of diseases. So it lets us make the link from diseases to genes. And therefore, if we can link diseases to genes, that means that we can also help resolve diseases by monitoring and editing the genes. And knowing what genes are linked to inherited diseases helps us understand them better and create effective treatments for them. Now, scientists can also look at genomes to trace migration patterns and look at how we've all descended from a common ancestor who lived in Africa, but now humans can be found all over the planet. The genome is mostly identical in all individuals, but as different populations of people migrated away, they gradually had different tiny differences in their genomes. And by investigating these differences, scientists can then work out when new populations split off. So, how can the genome tell us about history practically? Well, what it can tell us is migration patterns. Because if we all had one common ancestor, But then as humans started to migrate, they had tiny differences start evolving. That then tells you at the time and period in which they split off and what route they took as well. So now let's go back to the physics and maths tutor notes, because they then started looking at the DNA's structure. So how is DNA structured? Now, as we said, the DNA is the chemical that all genetic material is made from. So DNA is made up of nucleotides. These small parts called nucleotides. So each nucleotide has one phosphate molecule, one sugar molecule, and one of the four types of bases. So the four bases are A, T, C, and G. Okay. So one sugar, one phosphate, and one base. The sugar and phosphate molecules act as the backbone to the DNA strands. So the sugar and phosphate molecules alternate, and one of the four different bases joins to each sugar. Now, each base links to an opposite base. So A's always link to T's, and C's always link to G's. This is called complementary base pairing. So therefore, it's the order of bases in a gene that decides the order of amino acids in a protein. So let's just go through that in more detail now. So DNAs, these strands, these polymers, they are made up of small units, which we like to call nucleotides. Now, these nucleotides contain three things. These nucleotides contain three things: one phosphate molecule, one base, and one sugar molecule. And we're told that the phosphate and the sugar molecule form the backbone of the DNA polymer strand, and the base connects with this pairing. And also we have different bases. So A's always connect to T's, and C's always connect to G's, and this is called complementary base pairing. We were also told that it's the order of the bases. So the order of these bases determines the protein's function. So therefore the order of the bases determines the order of the amino acids, and the order of the amino acids determines the function of the protein. So going back here, we're told that each group of three bases codes for one amino acid. So a triplet of bases can create an amino acid, which then links the ideas we were just talking about. So three bases forms one amino acid. And we know that there are 20 different types of amino acids, and the order of this creates a certain protein. So this makes it easier now, because if we know that they're all linked, it's easier to look at this diagram and say, if the order of the three bases change, then that means that the amino acids will change, and that means that the protein will change. So it's all about the base, all about that base. You can use that song to help you remember. Now, it's important to remember that there are also parts of the DNA that don't code for proteins. These regions are called the non-coding regions. So we've got the coding regions of DNA, and then we've got the non-coding regions of DNA. So the coding regions of DNA are the things we've just talked about: the bases, the amino acids, the proteins, etc. But there's also non-coding parts and non-coding regions of the DNA. Now, what these are responsible for is that they switch genes on and off. Now, if you look at Punnett squares, this will make more sense because, as you know, it will be possible in a Punnett square to say you're looking at the blue-eyed allele and the green-eyed allele. So imagine if your blue was dominant and your green was recessive. So therefore you'll get blue, green, blue, green, blue, green, blue, green. And that then means that you're obviously going to have the blue-eyed because the blue eye gene is dominant. However, you also must understand that the green-eyed gene is still there. So even though you have the green-eyed gene, it doesn't show. And that's the main that's controlled by the non-coding regions, and that's how the non-coding regions also has a part in controlling what we look like and also our genome. It controls whether these genes show or not, whether they're dominant or recessive. So we could have a green eye gene and not have green eyes. We could have a blue eye gene and not have green eyes, not have blue eyes, etc. Now let's see if there's anything else we need to talk about. So we've talked about bases, we've talked about nucleotides, we've talked about complementary base pairings. Okay, so let's now talk about protein synthesis. So there's a couple of stages in protein synthesis. And protein synthesis, a lot of students get scared at initially, but do not worry, there's nothing to be scared about. I'm not going to show you this massive paragraph. I'm going to show you the broken down way that will definitely help you understand it. Firstly, what is protein synthesis? You may recognize the term from when you look at the purpose of ribosomes, which is to help with protein synthesis. But just going to what are the stages of protein synthesis. So protein synthesis, the purpose of it is that we want DNA to be copied and replicated when we're making new cells and things like that. However, not the DNA is too big to just be taken out of the nucleus and copied, for the process of producing a protein from DNA without having to copy it. Okay, there seems to be a bit of stream issues, so do tell me if the connection has gone or not. But yeah. So protein synthesis, the process of producing a protein from DNA. The process of producing proteins from DNA. That's what protein synthesis is. Now let's look at the first stage. So, firstly, the DNA that contains all of the genetic code for making a protein is within the nucleus, as is the DNA, but it cannot move out of the nucleus. So the first thing is the DNA holds the code for making proteins. But the DNA is too big to leave the nucleus. So that leads us onto our second point. We have this molecule called the mRNA molecule, and the mRNA molecule acts as a savior. So what happens is the DNA strands, because as we know, the DNA forms this double helix structure. So the DNA strands separate into two strands like this. That means that one side has one of the bases and the other side has the other bases. For example, if that was A, then that would be T. If that was C, that would be G. If that was T, that would be A, etc. So the DNA strands separate. Now, you then have something called the mRNA nucleotides. So the mRNA nucleotides, and these are the savior chemicals. So what they do is they look like these as well, these strands. So the mRNA nucleotides then go up to each DNA. Let's represent them by a different color so you don't get too confused. So imagine the mRNA is this color, pink. Now the mRNA molecules go up to the DNA strands that have now separated and form complementary base pairs to them. Now, as it's now formed complementary base pairs to them, what does this then mean? This then means that you have your mRNA nucleotides that are exact clones of the DNA. And that brings us on to the next stage. The mRNA nucleotide then join together to form an mRNA strand. And the strand then leaves the nucleus in the next stage. And as the mRNA strand leaves the nucleus, it then goes to the ribosomes. So this template of the original DNA has now made a copy and moved out from the nucleus through the cytoplasm, because it has to travel through the cytoplasm, to the ribosome. Now, as it has traveled to the ribosome, what happens is the bases of the mRNA strand, so imagine these bases here, they are then red. So the bases of the strand are red. Now, as these mRNA strands are red, what happens is you have, well, we have to go back to our original knowledge. Sorry about this. You have to go back to our original knowledge, in which we know that a triplet of bases codes for one protein, also known as one amino acid. Therefore, the bases are read on the mRNA molecule as triplets and therefore as amino acids, and then the corresponding amino acids are brought to the ribosome. So whatever is basically instructing the DNA, I need this amino acid, and this amino acid, and therefore the corresponding amino acids are then brought via things we call carrier molecules to the ribosome. And then what happens is these amino acids then connect together in a unique 3D structure to form a protein, and as it folds, it then has a specific shape, which we like to call the active site. So here you can really see your knowledge grouping together. Okay, now proteins have loads of different functions. So that's enough of protein synthesis. Let's move on to how proteins have the different functions. There's different ones, and you've definitely heard of them before, so it's great you don't have to relearn any content. So, proteins have different functions. Firstly, one function that could be is for enzymes. Another function is for hormones. And another structure is for structural proteins. So, when a chain of amino acids has been assembled, as we saw in the protein synthesis stage, it folds into a unique shape. This allows the protein to perform a task, whatever task that it's supposed to do. So there are a few examples of types of proteins. So you've got enzymes. These enzymes act as biological catalysts, help break down food, help with bodily reactions. Then you've got hormones. Hormones are used to carry messages around the body. For example, insulin is a hormone that's released into the blood by the pancreas to regulate the blood sugar levels. And you also have structural proteins. The structural proteins are physically strong, and what you would tend to think proteins are used for, they just strengthen the tissues in our muscles and things like that. Okay, let's now move on to the next thing, mutations. Mutations can occur when a gene changes, and a change occurs in a gene. One second, let me just check how many pages left of this topic. Okay. So occasionally, there will be a change in a gene. There will be a gene that mutates. Occasionally. Now, a mutation is just a random change, remember, it can't be predicted, a random change in the DNA of an organism. I shared with this slide now. So you can see mutation notes here. So a mutation is a random change in an organism's DNA. Sometimes mutations, however, can also be inherited. So mutations occur continuously. They can occur spontaneously. For example, when a chromosome isn't quite replicated properly. However, the chance of mutation is increased when you have exposure to certain substances, such as certain types of radiation. Now, mutations change the sequence of DNA bases in a gene. This then produces a genetic variant. A genetic variant is something that's different from other genes. Now, as the sequence of DNA bases code for the sequence of amino acids that make up a protein, mutations to a gene can then lead to a change in the protein. It doesn't always, but it can. Now, as you can see in the three ways on the screen in which a base can be changed, causing a mutation, either through inserting it, deleting it, or substituting it, this can have a massive effect on the protein. Now, most mutations have very little to no effect on the protein. Some will change it to a small extent that its function or appearance is unaffected. However, some mutations can have a really serious problem and affect a protein. They will code for an altered protein that will change its shape, and this could affect the ability for that protein to perform a function. For example, if their enzyme's active site is changed, the substrate would no longer be able to fit on it or bind to it. Or with structural proteins, they could lose their strength, meaning that their shape is changed and it's pretty much useless. If there's a mutation in the non-coding DNA, it can alter how genes are expressed. So, insertions. Insertions when new base is inserted into the DNA sequence that shouldn't be. Now, remember, every 3 bases in a DNA-based sequence codes for an amino acid. So an insertion means that every amino acid after that will have a knock-on effect because they'll be read in the wrong triplet. Deletions is when one is deleted. Like insertions, it can have a knock-on effect. However, substitution is when they're just swapped or changed to a different base, and this might not have a long, massive effect. Okay, now moving on to reproduction. So just going back to the beginning of the notes here. Sexual and asexual reproduction. So sexual reproduction is where genetic information from 2 organisms, a father and a mother, is combined to produce offspring with genetically different to either parent. So this is really important that we get this definition right, because we saw that in paper 1 of 2023 biology exams, sexual and asexual reproduction did come up, but they didn't want you to talk about it in the context where we're talking now. So remember, sexual reproduction and asexual reproduction. So sexual reproduction is when you have genetic information, so genetic info from two organisms, both the father and the mother, and this is then combined to produce offspring which is genetically diverse and different. So genetically different offspring. Now, in sexual reproduction, the mother and father produce gametes by meiosis. So you can see the process of meiosis in the notes here. So meiosis is the formation of 4 non-identical cells from one cell, whereas mitosis is the formation of 2 identical cells from one cell. So meiosis is how sex cells are produced, egg cells and sperm cells. In humans, each gamete or each sex cell contains 23 chromosomes in comparison to every other cell in our body, which has 46 chromosomes. So, therefore, gametes only have one chromosome, whereas as we know, because chromosomes exist in pairs, that's why we have 46, because it's 23 pairs of chromosomes. Whereas in a single cell, it will only have 23 singular chromosomes, not pairs. Now, the egg and sperm will fuse together during fertilization in sexual reproduction. So, sexual reproduction is where genetic information from two organisms produce a genetically different offspring. It involves the fusion of both male and female gametes, and there are two parents, so there's a mixture of parent genes. This is why the offspring inherits features from both parents, and the mixture of genetic information provides variation. For example, flowering plants can produce in this way too, because they have egg cells, but their version of sperm is known as pollen. Okay, asexual reproduction produces genetically identical cells. So this uses mitosis. So sexual produces meiosis, asexual produces mitosis. So this is when you have offspring that is genetically identical to the parent because there's only one parent. There's no fusion of gametes, there's no mixture of chromosomes, there's no genetic variation. All exactly the same as a parent. So you see organisms such as bacteria and plants and some animals that reproduce asexually. Now, you can see the process of meiosis here. So the cell makes copies of its chromosomes, so it has double the genetic information of a normal cell, and then it divides into 2 normal cells, and then it divides into 4 gametes. And they're all genetically different because of a process called genetic crossing. So before the cell divides, it duplicates. Its first division by meiosis as the pairs line up. The pairs are then pulled apart, so they only have one copy per chromosome, and some of the father's chromosomes are shown. And then in the second division, the chromosomes line up and the arm of the chromosomes are pulled apart again. Now, the cell produced by gamete fusion replicates itself. So after the two gametes have fused during fertilization, the resulting new cell divides by mitosis to make a copy of itself. As the embryo develops, these cells then start to differentiate into the different types of specialized cells. Now, there's advantages and disadvantages for both sexual and asexual reproduction. You can see through this table here. So advantages of sexual is that there's variation, which could provide a survival. Individuals may die. It will prevent all species from becoming extinct. It allows selective breeding. It also ensures that organisms with different desirable characteristics can be bred to produce offspring with even more desirable characteristics. And it also speeds up the process of natural selection. Advantages of asexual reproduction is when there's only one parent. It uses much less energy, it's much faster, and in favourable conditions, lots of asexual reproduction can occur. So, some fungi can produce asexually, and they can produce sexually as well when the conditions change in order to increase variation and avoid extinction. So, if you have strawberry runners, they produce runners which allow them to grow. Now, malaria is another example of something that reproduces by both methods. So it's caused by a parasite, and when a mosquito that is carrying the parasite bites a human, the parasite is then transferred to the human, and the parasite then reproduces sexually when it's in the mosquito and asexually when it's in the human. So sexually in the mosquito, asexually in the human host. Okay, now moving on to X and Y chromosomes. So there's 23 pairs of chromosomes in every human body cell. 22 are matched pairs of chromosomes that just control characteristics, and the 23rd pair, labeled XY or XX, these are the chromosomes that decide upon your sex. So let's find the notes on that here. Here, okay. Sex determination, the 23rd pair. So the 22nd pair are all matched pairs, whereas the 23rd pair is different. It depends on whatever you have, X and Y chromosomes, whereas you have two X chromosomes. So therefore you have the Punnett square, which can be seen here, and it produces exactly a 50% chance of you being male and a 50% chance of you being female. So it all depends. Now, you also have other methods to look at genetic diagrams which show the gamete combinations. So you also have another type of genetic diagram. It's more complicated and it's not shown on this notes, so I might just skip over that for now because it's not too necessary. So let me actually show you genetic diagrams because I want to cover everything and make this a perfect video for you guys for your exams. So let me just if you find this video helpful, do be sure to tell me. I'll massively appreciate it. Okay, I found a genetic diagram example. So here you can see what a genetic diagram looks like. So what they do is they basically show all the possible alleles of offspring. So it can also be represented through a Punnett square, but sometimes you may be asked about this, and this year they seem to be a bit unpredictable with the exams that they're setting, so just be aware of all of the different methods. So as we know, different genes control different characteristics. Some characteristics are controlled by a single gene. For example, the fur color of a mouse or the having color blindness. It's controlled by a single gene. However, most characteristics are controlled by loads of different genes working together. So we have two versions of every single gene. Therefore, a version of a gene is called an allele. So we have two alleles of every gene, one from our mother and one from our father. Now, if two alleles are different, only one can determine what characteristic is present. So the allele for the characteristic that's shown is called the dominant allele because it shows you. And you use a capital letter, whereas the one for that's not dominant is called recessive because it's not strong enough. So in this diagram below, you can see that capital F, and this is talking about cystic fibrosis. So capital F means that it's a dominant cystic fibrosis gene, and the lowercase f is a recessive allele that causes cystic fibrosis. Now, for an organism to display a recessive characteristic, that means that both the characteristics have to be recessive because dominant overrules anything. Your genotype is a combination of all of the different alleles that you have. So your alleles work at a molecular level to determine what characteristic you have, your phenotype. So, in plain English, your genotype, the genes in you, determine your phenotype. So at the top of this genetic diagram, you can see that there's the parents' genotype here. Well, okay, let's break this down for you. So at the top of this diagram, you've got this line here. This shows the parents' phenotype, the physical characteristics. You can see that one parent is a carrier of cystic fibrosis and another parent is not a carrier. So they're both not a carrier in this case. They're both not a carrier. They both do not have the disease, but they are carriers of the disease. So in a genetic diagram, the first line will always be the parent's phenotype. Then the second line will be the parent's genotype. So this is the genes that they have. And the genes will reflect the phenotype because it's type. So here you can see that they both have a dominant allele to be normal and a recessive allele which highlights that they're carriers but they're not affected by it. So that's the second line. Then the third line shows the gametes genotypes here. So the offspring, what do they have? So you can just show all of the possible letters. You can see the two alleles that the parents can give. And then in the final line, they have created all of the possible different endings. Now, a way we would represent this in a Punnett square, for example, now Punnett squares are arguably much more simpler than this. So what you have in a Punnett square is it will look like this. Let me make it bigger. Okay. And on one side, you have your first parent, which in this case is FF, so capital F, lowercase f, and in this case, the other parent has the same genotype, so it's capital F, lowercase f, and then we don't represent this line. We just move straight onto the offspring genotype, so you can have capital F, lowercase f, capital F, lowercase f, and lowercase. And that all of these things here represent the genotypes of the offspring there. So you can see how it's equally represented. Okay, so this has gone a bit over time and we're only on the second topic, so I may have to pause this stream here or pause it earlier so that you guys can have a break because definitely 2 hours is a long time. But very well done. Let's complete this topic, and then what we'll have to do is we'll have to move on to another topic, maybe tomorrow finish it off. Or maybe later today we'll finish it off. It's up to you. Do comment how you're feeling about that. But let's just quickly finish off this topic. So, inherited disorders, let's quickly go over that. So, inherited disorders, you need to know that genetic disorders are caused by inheriting certain alleles. So, cystic fibrosis is a genetic disorder of the cell membranes. So, what happens is it results in the body producing lots of mucus, and this sticks in the air passages and in the pancreas. Now, the allele that causes it is represented by the lowercase f, and because it's recessive, as we looked at in the Punnett square and in the genetic diagram, people with only one copy of the allele won't have the order. So both parents have to have cystic fibrosis in order for the kids to have it. in order, yeah, because it's recessive. Polydactyl is actually caused by a dominant, so you only need one parent who has polydactyl to pass it on, and it just means you have extra fingers or extra toes. It isn't necessarily life-threatening, and if one parent has it, then it's a 50% chance of you having it. Now, embryos can be screened for genetic disorders. So embryo screening, it happens during IVF as well, and the genetic disorders can be detected, and you can also get the DNA from an embryo in the womb, as well as the ones that are going from a lab. However, there's ethical, social, and economic concerns because it's quite controversial. The decisions that it can lead to, termination, just because they have a disability, that's lots of ethical problems arise from that. Now, people who are against embryonic screening say it implies people with genetic problems are undesirable. And it also comes to a point where it could lead to things such as designer babies, where you're only allowing people with certain characteristics to be alive, which is definitely wrong. Now, let's go on to variation and evolution before we have a quick break. So, variation depends on two things: the genotype, which is the genes it inherited, and the environment it lives in, which is the place it lives in. So genotype, genes are passed on from the parent in the sex cells. The combination of the genes from the mother and the father create genetic variation. Only identical twins have the same genotype, and there's lots of genetic variation in a population. So the environment it takes place in, the conditions the organism grows and develops, also affects the appearance. So a lot of characteristics come from a combination of both the genes and the environment it lives in, but it all depends in the end. Now, okay, most characteristics are due to both of them. So as we said, most characteristics are a culmination of both genes and determined by environment as well. For example, the height that an animal or plant could grow is determined by its genes, but also whether it grows that tall could be dependent on the environment. For example, if you don't have as much access to food, you could have stunted growth. Now, mutation is also um, relevant in this section as well because of the thing that mutations, as we said, changes to sequences of bases that are totally random and spontaneous. So, changes to sequences of bases in the DNA. And most mutations have no effect at all on the protein that the gene codes for. So most mutations have no effect and small influence. However, some have more of an effect and could lead to a selection pressure and survival advantage. And that leads us on to evolution. So evolution is that all of today's species have evolved from simple life forms over billions of years ago. Okay, I don't want to rush this topic, but it has been almost 2 hours, and I do want to give you guys a break. So what I'm going to do is I'm going to come back to this probably at 12 o'clock or half 12. So hopefully you can join me back there, and I will complete biology paper 2. Thank you so much for tuning in, and see you very shortly. Have a good break.